working with plant morphology and plant morphology and plant anatomy from some species of cacti, especially species of the tribe Hypsalidae. And today I'm gonna present to you the flower of some epiphytic cacti, an structural view, and I expect to show you guys a brief overview of the cactus flower, some implications of the floral morphology in the epiphytic cacti, much related to the reproductive biology, with some examples in the Iloceri and Hypsalid tribe. And if you got time, some details of the floral anatomy and some perspective for the future. It will be pretty much a descriptive talking, and I'd like to apologize you guys for my bad English. If you do not understand, Tanya, feel free to interrupt me to make it, everything clear if it's necessary. So, as we know, the cactus family has a family from the New World, as the Bromeliaceae family, or other example from some hummingbirds. But there is some, there's one species and some farms that occurs naturally out of the Americas. Hypsides baxifera that occurs in Africa, Madagascar, Sri Lanka. There are some hypotheses that how this species has reached there, but what has been reported at these farms that occurs in that old world, they are the morphology are a little bit different because they have passed for recurrent cycles of the polyploidea. So the farms in that specimens that occur in there, they are polyploid, and they are morphologically some difference between the species from the Americas. And then when we look at the center of the diversity for the cactus family, there are some regions, some spots that has more genus. The descent of the diversity is a little bit more higher, as in the northeast coastal zone in South America, in Brazil, in the Andes Mountains, in Mexico. And when we look at the risk for the family, also this region are suffering some kind of risk. And the epiphytic cacti from the Hypsalid tribe occurs mostly in South America, and Iloceri tribe that occurs mostly in Central America, they are suffering severe risk as well. I cannot tell about the risk in the Central America or Mexico, but I'd like to show you some here from Brazil. The epiphytic cacti from Hypsalid that occurs in Brazil, they occurs in the Atlantic forest. Is a very humid environmental, and they occur as epiphytic species. And in that region is the place where the most population of the Brazil lives. The big cities are in that region. So if you look at when it was before the covering of the forest in that region 500 years ago, and compare what left today. So in green, that's what we have keep in forest. And if we consider every single green dot here, we still have about 12 to 14% of the Atlantic forest remained. And all where the green guns are big cities or even big field crops. So not only the epiphytic cacti or the species from this biome are really in danger at all. And then the examples I'll try to show you today about these two tribes that traditional were included epiphytic cacti in Hypsalid that mostly are in the South America, and the tribe Iloceri with most of the species are in Mexico, Central America, and Caribbean. Though there are some Hypsalid species who lives here, and quite a few species of Iloceri who occurs naturally in the South America too. And it's worth to remember that after the Nadia Skorotskovas paper, the two genera, Dimia and Strophocactus, were excluded from Iloceri and included in Echinoceri, but it is a whole other kind of discussion for other time. And before I try to show you the results, I'd like to calibrate the structure of the flower for the cactus family. Although some species of cacti have flowers with semi-inflow or even a superior ovary in the flower in the ancient lineages, like the Pereskia, Leonbergera, the most flowers in the family has inferior ovary. And then we have the eye structure called pericarpel, which are involving the ovary itself with the ovals. And in the top, usually we found a nectar structure, and then some lineage will have a bigger or not so bigger floral tube and the perianth with tepals, and the stamens, and inside the stigma and the style. When we compare to a regular flower or hypothetical flower with a superior ovary, it would be more or less in a very, very simple thought that at this the ovary went down in the receptacle and the peduncle tissues. So when let me go back here. Look at the bottom of the flower in the cactus species, we will not find a peduncle or a receptacle as in the other flowers that has the superior ovary. And it's make the structure in the flower a little bit hard to understand sometimes. Book in the 60s has many papers that he got the conclusions about how the steps of the evolution in the ovary in the family, working in Pereskia and Berger species. Ingrid Ruth in 1977 has a nice book in review about these possibilities, but there is one evidence, the anatomical evidence about this ovary that went down in the receptacle, that are the recurrent vascular bundles or the opposite vascular bundles. So when this structure of the ovary went down, the vascular bundles follow it. And then when we look at in the cross section, we have, for instance, a regular vascular bundle with xylem and phloem, and in the other side, the opposite vascular bundle with the xylem and phloem swift deposition. So it's not common to see that in angiosperma. That is not exclusive for cacti, but it's still rare in the angiosperma. So now I try to show you the profile of the floral morphology in some cacti species. I will start for the tribe Iloceri, and then go to the Hypsalid, and then see if I can make me clear for you guys. This species, Gisocactus ackermanni, has a beautiful, beautiful flower. It's one of few species in Niloceri that has flowers in color. Is a beautiful red color, and that has the anthesis during the day. Most of species from Niloceri, they are queen of the nights, and the anthesis lasts only one night. This nice flower of Gisocactus ackermanni has the same pattern of the flower I showed you before, the Brasilopuntia, but it has a floral tube that it's quite long. And in the base part of the flower, we have the ovary, and then we will have the floral tube and the perianth with the androecium and part of the gynoecium. In the tube, we have about 2/3 of this tube with in the internal structure of secretory structure, the nectary or the nectariferal tissues. And it produces large amount of nectar and a diluted nectar in terms of water content. The other third part of the tube is the part when the filaments of the anthers are inserted. And when we look at the surface of the nectary of this nectar chamber or nectariferous chamber, we found lots of trichomes in the epidermis and also some stomata for the nectar release. Another flower, all flowers are beautiful in these tribes, but this the Selenicereus cetaceus, it's quite different when compared to Gisocactus. It's a whitish flower, a huge flower, more than 20 centimeters long, and more than 20 centimeters diameter too, and it's still the same pattern. This long tube has at least 2/3 of secretory tissues, the nectary, that also has in his epidermis trichomes and stomata to release the nectar. The difference between the other flower is this flower of Selenicereus cetaceus has hundreds of stamens that produce a lot of pollen grains, along with lots of nectar. Lots of nectar is any amount that is over 100 microliters. And for the tribe, as we could see, this is the pattern of the flowers. The tube would be a little bit longer or less, but still a long tube. Most of these species has flowers that has the anthesis during the night, and the secretion, the release of the nectar is made by the trichomes and by stomata. This stomata depends of the author, could be only called stomata or a functional stomata or nectar stomata or a pore, but anyway, there is no functional stomata here to release the nectar. I'd like to call attention for this species, Selenicereus antonianus. It's quite common here in Brazil as ornamental. Is not from here, was introduced. The plants here, they bloom, they produce flowers, but they never go to the fruit set. So there is as far as I could know, there are very few information about the fruit biology of these species, besides be very common in the market and the markets as an ornamental plant. And when you look at the Hypsalid tribe, they are a species who lives as an epiphytic habitat. Some species has vegetative organs similar to other species of Iloceri, but the flower has nothing similar. They are very, very small, tiny flowers when compared to Iloceri, that has the biggest flowers in the family. And here we go to the tribe who has the smallest flowers in the tribe. Pardon, I'm sorry. The flowers in Hypsalid, they are more rotate or bell shape or bow shape, but there are only one genus will find some tubular head structure. And the real words besides the pollen, the nectar, it's different because the flower size, it's very tiny. So the structure follow this this size, and this hypsalisteres. We got the ovary inferior, surrounded by the pericarpel, but the nectar structure in the hypsalid, they are pretty much like in annular shape, looks like a donut, and it is linked in this region. It's like in the tube, like the micro tube, a very, very reduced the tube when it's bear the structure of the filaments, the stamens, and the secretory structure of the nectary. Other species has a different flower morphology here and there, but they still small flower, and they have most of them annular shape of the nectary. Only some species that has flowers like Hypsalis cereuscula, that the annular structure of the nectary is much more merged in the tube, and this species has a tube that's a little bit longer when compared with other Hypsalid species, but for the family is still very, very, very short. And all the species we could analyze of Hypsalid, they release the nectar through stomata. There are no trichomes in the surface of the nectary. Other genus in the Hypsalid family, like Achiora and Lepismium, they have some difference and variation in the flower morphology between the genus, and it follows the morphology of the nectary. In Achiora gartneri, for instance, it has a rotate, a little bit bigger flower, about 5 centimeters diameter, and the nectar is more like the ball shape. In the Lepismium genus, both Lepismium cruciforme as Lepismium vermigiano, the morphology between the species of the flower are different, but the shape of the nectar, they are pretty similar, much more like in a bell shape, and also release the nectar through the stomata. And the Schlumbergera is a very interesting genus in the cactus family, and even for the Hypsalid, it has been some change that was considered Schlumbergera traditionally. But even though we have species like Schlumbergera truncata that has one of the largest flowers of Hypsalid in this tribe. It has a flower that has a tube, but the nectar itself, it's a little bit similar with those from Lepismium or from other species of Achiora. Here it's bell shape, but some species of Achiora, Schlumbergera, they have in the top of the nectar like a roof. And this makes some flowers, the flowers of some species, very, very unique, because it has a nectar, a nectary, like in a bell shape, but it has a roof, and it's a different, forms a different kind of a chamber for keep the nectar. And the nectar is released by stomata as well. Okay. In general view, the flowers we analyze in different genus from Iloceri. Ilocerius, there is no more, it's Silencerius now, but they have a vast nectariferous tissue. It's classified as a chamber, is a nectar type as a chamber. Using the classification of Buxbaum in 1963 or the 60. And in this paper, in the work of Buxbaum, it was one of the pioneers in the cactus morphology. He has classifications of the different types of nectary, and he separated some chambers in nectar type of open chamber or diffuse chamber. And the difference in the open and the diffuse is how is the arrangement of the stamens in the top of the tube. But as I could know, this is not easy to follow because there are some variations inside the same populations, mainly in the species who has huge flowers with hundreds of stamens. But a chamber is still a pattern in all genus of the tribe, as we could see. In Hypsalid, there is a kind of more variation in the morphology of the flower and also in the micromorphology of the nectary, since we have much more species of Hypsalis. It's easier to find a nectar annular shape in the Hypsalid. But Achiora and Lepismium and Schellenberger, they have different kind of nectar because they have a floral tube, a kind of different than the Hypsalid flowers. And some species of Schellenberger, not all, but some species has that roof that he call a subtype, like a nectar holder. And we have the opportunity to look some qualities of the nectar too. And the trend we found was Hypsalid, the nectar found, the volume is less because of the size of the flower, but also the sugar concentration is very high. The flowers, they are more rotate and the nectar are more exposed. So many features of the flower contribute to the concentration of sugar in the nectar solutions besides the productions inside the flower. Iloceri, on the other hand, has large flowers with long tube, and many species has nocturnal anthesis, and then the nectar has a different kind of exposition when compared to the Hypsalid. And the nectar quality in terms of the sugar concentration, it's is much more diluted, as the concentration of sugar is much more diluted than in Hypsalid. And it has been known that the quality of the nectar are related to the diet of the visitor, or the ability of the visitor to feed on the flower or take the research as the nectar, for instance. And that species of Schellenberger genus that has a tubular flower, it is an intermediary flower between Hypsalid and Iloceri because it has a diluted nectar with inside the nectar chamber. So this is different both from the Hypsalid and Iloceri. So taking the Schellenbergia truncata as an example, this kind of flower, the floral display, the color, and we could expect it to see birds visiting this flower, because it's the features of the flower fit very well in ornithophilos syndromes of pollination. I have seen a couple times birds visiting the Schellenbergia flowers, but they are only, and there are some notes here and there that hummingbirds are pollinator of these flowers, like Christmas cacti, but they are only personal observations. As far as I know, there is no records yet, because to promote the fieldworks observations in these plants, as the Schellenbergia or other epiphytic cacti, is sometimes very complicated and very tricky. So it's not easy to found the plants. Sometimes the population there has no many individuals to have a satisfactory analysis. And then there are no many information about that still. Talking about the expected pollinations, this we could see and make the observations is Silenicereus cetaceus here in the Minas Gerais state from Brazil. And this species has a huge flower, as I have showed before, with nocturnal anthesis. And then we expected to see this flower being visited and pollinated by nocturnal animals like sphinx, hawk moths, and even bats. But in 3 years of observation by my student of PhD, he was able to see only one single visit of a sphinx. And this flower has a huge amount of research, millions of pollen grains, and lots of nectar is produced, and but no nocturnal visitors at that region. But it's producing fruits. And then we were discussing the first thing we thought that they are self, in many species of cacti, they can reproduce by themselves. They don't care about the others. They are self-compatible. But for my surprise, because it has been recorded for other species in Mexico, there were other animals visiting these flowers in daytime. So the honeybees were taking advantage of the very beginning of the anthesis before the sunset, and were entering in the flower and take the pollen, collecting the pollen, and then walking in the flower and promote both self-pollination and cross-pollination. And at night, there's no activity of the bees. But in the next day, in the first sunlight, about 5 a.m., the honeybees get back to the flowers that are finishing their closing, finishing the anthesis, but they went there collecting the pollen that still be available in that flower. And at this time, the stigmas is still receptive. And so these bees are able to self-pollinate or cross-pollinate the flowers. And so we have some experiment of exclusion, and then we conclude that these populations is self-compatible, but the flowers that were self-pollinated by hand produce less fruit with less seed. And the flowers that were visited by the honeybees, they produce more fruits with more seeds. And also these species are able to produce fruit to are self compatible spontaneously. But anyway, these honeybee are playing a role to guaranteeing the reproductive security of these populations of Solenis cellulose cetáceos in that region. Because in the absence of the nocturnal visitor, there are many hypotheses how the moths are being suffering in this time close to cities or other many issues that are impacting the life of these insects. And we could see that there are a small shift in the flower phenology to keep the reproduction of these species, although the floral morphology is still features of a specialized flower, a very specialized flower. Another visitor of the same species was some ants that were in the flower, mainly during the development of the flower in the floral bud phases, and they were collecting and feeding the nectar in the scales in these extranuptial floral nectaries that release the nectar to stomata. Now I will show you very quickly, to not take many of your time, about the anatomy and some aspects of the anatomy in the floral, in the flower of some species of Iloceri and Hypsalid. I'll start with Epiphyllum. Epiphyllum is a very, very didactical cactus flower and has a very long tube. And when you look at the anatomical structure of these flowers, we have in the base of the tube, half of this tube is nectary. In the bottom, we have the ovary. In the bottom, of the region of the tube, we have in the internal part of the secretory tissue. And then the pattern is the same, the trichomes and stomata to release the nectar, secretory parenchyma, and a huge network of vascular bundles. But these vascular bundles go up, and they are not vascularizing only the nectary. They go to vascularize every every filament of the anthers. And when you look at the base of the flower in the ovary region, as I said before, we know that the external part is the pericarpel, the internal part is the carpel itself that bear the ovules. But the boundary, the anatomical boundary between the pericarpel and the carpel itself is very complicated to say where finish one and start another. We know that there is a kind of ring of huge vascular bundles that promoting the ascendant system that comes to the stem. But when finished the pericarpel, that is a receptacular tissue in origin, and start the carpellar tissues, it's very complicated to sell. All right. And this is another point in the cactus flower that I think it's very common for the entire family. The anthers, they could be bigger or smaller, depend of the flower morphology, but the anatomical structure, I think, is pretty stable in the entire family with 4 layers, epiderm, the second layer responsible for the opening of the anther, the endothecium. The third layer is called middle layer, and the inner layer, the tapetum, the secretory tapetum, that provide the microsporocytes. And this structure of the anthers, I think it's a pattern for the entire family, not only for the epiphytic one. The ovals in the Iloceri species of flowers are huge, so it's common. Our cactus is an example for Circinotropous ovals. They're not common in the angiosperm, but more common to find Circinotropous ovals in the cactus family and mainly in the Iloceri tribe. When you go to the Hypsalidae and the anatomy of the flowers, it's of course it's different because of the morphology of the flower is different, but the pattern is pretty much the same. We have the external part of the pericarpel, and we have this kind of network of the ascendant vascular bundles, and these bundles have some ramifications that goes down to form the recurrent systems or the opposite vascular bundles that vascularize the ovary. And we have other series of ramification that goes to the both stamens and also to the stigma. But if we pay attention here in these cross sections of this flower of Achiora salicornioides, in the region up here, close to the nectary zone, up to the ovary, we have many vascular bundles that are coming up, and these vascular bundles are vascularization both the stamens, the stigma, and the tepals. And the same vascular bundles that will vascularize will go to the stamens, the filaments, they are vascularizing the nectar too. So many species we have looked at the anatomy of the flower, they share the vascularization and the nectar and the filaments. There are quite a few species, or only a few species, that has some difference in the way of the vascular bundles goes, and sometimes it has a vascularization in the nectar, as in Schumbergia truncata, for instance. But usually they share the vascular system between the nectar and the androecium. It doesn't matter if there is a long tube flower or a very short tube flower, as in the Hypsalis. This Hypsalis cereuscula, for instance, this is a floral bud in longitudinal section. We have that pattern with the external pericarpel, the internal carpel, the ovals. Here, the ascendant vascular system that goes, and just open in the top of the ovary. It goes down to make the vascularizations of the ovary, but has the ramification that goes to the stigma, and a step above it goes to the filaments of the androecium, and it's passed to this dark region here. It's the beginning of the nectar, and other vascular bundles goes to the tepals. If we try to make some comparison between these epiphytic tribes, Iloceri and Hypsalidae, we have different, I'm sorry, in the oval morphology. For instance, in Epiphyllum philanthus, the funicle is long, but is unique. In the Selenicereus undatus, for instance, the funicle are ramified, and the number of ovals per placenta is much higher. In Hypsalid, for the other side, we don't have circinotropous ovals. The most of ovals are anatropous, or in some species, they have a little projection of the placenta here, but they are tiny when compared to the Iloceri. Another aspect I'd like to call attention besides the ovals, here the anatropous ovals of Hypsalid species, is about the tepals. In the first genera, we have analyzed in the scanning microscopy. There are some like trichomes or some cerds in the very top of the tepal in Hypsalid, and Iloceri, they are very, very glabrous. Another point of their difference between the structure in the flower is in the style. The style in the Philomphilanthus, for instance, the transmissor tissue is solid inside, but in Selenicereus cetaceus, Selenicereus undatus, it is, has a space inside the style and with an epidermis. That's the most I have selected to show you. And then for the future perspective, I'd like to summarize all the data we have collected for the last years and see if we could have a view a little bit more interesting in view of the evolution of these lineages. And I can tell it's more, the difference are more relevant in high level, between tribes, between genus, between species. It's there are less difference in the internal structure of flowers. It's kind of expected anyway. And the reproductive biology is still an open field. There are some people studying. There are some literature about that. But even for the native species, there are lots of to understand this still. And the epiphytic cacti, they are an important component in the community, in the plant community, because sometimes they provide research for animals out of the season. And the use of other and modern technologies that are more accessible today is not in every corner. You can find a CT microscopy, but today is easier to do this analysis than it was 10 or 20 years ago. And it could help that this field of the plant morphology, plant anatomy, that could expand lots of things, go faster and have the results and even the analysis of these results. I cannot finish it before thank you my collaborators that has been working with me for a lot of time, and also I need to thank you to my students that are helping me to keep going. And so that's it. Thank you very much, everybody. I'll be glad to, if you have any questions.
Thank you very much, Odair, for your presentation. Such a wonderful, beautiful pictures you showed to us today.
Thank you.
If anyone wants to ask a question, please go ahead and unmute yourselves, or you can also raise your hand. I don't see everybody in my screen at the same time, but you can feel free to just unmute. If you are more on the shy side, you can just type in your question in the chat and we can read it for you. So let's give a minute for people to think about it.
It's fine.
Well, in the meantime, I do have a question. Odair, you led till the end for your future activities you want to do to discuss about the evolution, but it's inevitable to think what do you think about the evolution of these nectaries and this particular floral type? Especially I'm thinking in contrast with other cacti, regular non-epiphytic cacti. So is it in response to pollinators or the particular ecological conditions in more tropical areas?
So it's very a very exciting topic because as a floral research, it's suffering the pressure of the pollinator that could drive some change. But in the same time, there are some characteristics of the nectar that looks like, for instance, in the hypsalis, there are only stomata in the surface. In the Iloceri has stomata and has trichomes. But the flower of Iloceri is, if you look at the morphology, is pretty much similar from the columnar cacti or the terrestrial cacti that are close sided. And that flowers, there are no trichomes. They are more with stomata. And then I think if it's we'll be able to join a matrix of the results and analyze it in the perspective of the evolution, it could be interesting, even if that groups that has some doubts, it could be given some light. Will not be the golden bullet, but could be one more light to be turned on.
Yes. Thank you. I think we have questions. Joseph and then Andreas, please.
Yes, hello. Thank you, Odair. Very beautiful work. So I have a question related to the nectary release through stomata. I'm not working with flowers. So is it very common for flowers in general, or it's some unique features of cactus flowers?
Hi, Joseph. It's nice to see you here. Thank you. In cactaceae, is common to expect the release of nectar by stomata. It's a functional stomata. It's still pretty much opened because they produce lots of nectar. But in flower in general, it's very variable, because sometimes it could be a secretory structure that has an epidermal secretory, or sometimes there are some trichomes specialized to produce and to secrete the nectar. Other times the cells of the epidermis secrete the nectar and it's still between the cell wall and the cuticle. So in angiosperma, it's very diverse. But in cacti, I would say, as far as I could see, it would be the two options, or a trichomes or stomata. Because even in nectari that are not related to pollination, as in the scale, we detect some stomata. We interpret that the stomata will release the nectar, but we will not able to find a parenchyma, a specialized parenchyma, or the cells that are producing the nectar, because in the floral nectar is vast, the parenchyma that producing the nectar. But in the scale, we couldn't see the cell are producing nectar or identify very well, but the stomata is still there.
Okay, but in other families, is this, can you find this same phenomenon?
Yes.
Okay.
It could be found, but it's not mandatory.
All right. Thank you very much.
All right. Thank you.
Thank you, Joseph. Andreas, please go ahead.
Yeah, first of all, obrigado. Thank you very much for this very interesting presentation with the nice pictures. So for me, I got new insights about the morphology. I do not have a question, more two remarks, what I would like to make to something what you mentioned. You mentioned that Cereus antonianus didn't produce fruits. I could produce a fruit with a cross with Akamani hybrid and produce a lot of seeds, and they were viable. So I got a lot of plants. Unfortunately, I don't have anyone any longer, but for me it worked. So the fruit was a little bit bigger than the table tennis ball. So it worked.
Nice.
Maybe you can try that.
Learn that. Yeah, I try here, try to do some crossing. The problem here in Brazil, that's the Cereus antonianus, they are pretty much lonely, and it takes several years to bloom. But I try with the ones I have in my greenhouse.
Yeah, I hope that it would work for you as well. And the other thing what you mentioned, so that honeybees could be a good pollinator. So this year I cultured my cactus collection outside in an open area because I hadn't a greenhouse for a certain period. And there are a lot of plants that I tried to pollinate by hand under defined conditions. I was not successful. And this season, I got a lot of fruits on these plants. So either pollinated by honeybees or by bumblebees. So that could be a good pollinator. But of course, then you don't know the parent.
Yes, the honeybees, they are very good in taking pollen, but there is a kind of paradox here we could not don't tell about that, because in America they are introduced, and also they are very good in taking pollen and make the pollination, but also they are good in to remove the native species, and this is another whole problem. But in terms of the collecting pollen and transporting pollen, they are very good, no doubt. But in terms of the ecosystem, sometimes they are not so good in terms of the native bees.
Thank you, Andreas. Thank you again, Odair. Congratulations for your presentation. Somebody here asked if there's a place I could watch a recording. Yes, we are recording, and we eventually are going to upload in our YouTube channel. Stay posted for that. We are a little bit slowly, but we are getting there. We are some announcements. We are requesting for volunteers. If you would like to help us organizing these seminars, please feel free to email us, and we can interview you. Well, thank you very much, Odair, again. Thank you, everybody, for joining us, and see you next time.
Thank you, everybody. Thank you, Tanya. It was my pleasure.